Hello, everyone, and welcome to our webinar entitled From Regional to Global, the FIP Latin America Biowaiver Project. My name is Giovanni Poletti, and I have the privilege of serving as the FIP Scientific Secretary and also today serving as the moderator of this session. We hope to have a great crowd here, and I know already see some wonderful chats welcoming all the people across the globe, wherever it is, whether it's morning, afternoon, or late evening. We welcome you to this particular event. Please let me address a couple housekeeping rules first. For those who have been participating in FIP seminars or webinars before, you're already aware that this webinar is recorded as well as live streamed via our YouTube channel. Then we also have the ability to store those webinars. And so if you want to look at them later on or share them with some friends, please direct them to the website at www.events.fip.org. Throughout the presentations, we will collect questions in the Q&A box. So please, if you have any questions related to the topics that are presented by our speakers, feel free to add your questions in the Q&A box and we'll address them during the panel discussion at the end. We're also collecting feedback on our webinars. So if you have any suggestions, feedback, please feel free to send those to webinars@fip.org. And finally, as a benefit for our members, we also encourage you to consider membership in FIP if you're not a member yet, and you can see the different options of membership in the website listed there below. I want to acknowledge first a couple people who make this event possible, people who you don't see in this particular presentation. They are members of the FIP team that facilitate the digital events. So thank you for helping us out with the technology here. And then particularly relevant for this project is the steering committee of the Latin America Biowaiver project. And we have a few representatives of that committee participating in today's webinar. Last but not least, I want to also acknowledge the support of our external partners, the innovation of Biopharmaceutical Evaluation Centers, Indus Pharma, and Legis. Before we begin with our program today, I also want to share with you that we have opened the registration and the abstract submission for FIP's 81st World Congress in Brisbane, Australia, this fall. So please consider submitting your abstract before 1st of May and register before 15th of June for this special early bird fee. And you can find more information about the Congress on the website at brisbane2023.fip.org. With that, I'm going to start a brief introduction of our webinar today. And it is important to understand that FIP recognizes the needs, the different needs of the regions. And we have now started with this project a slightly different approach than in the past, where FIP can actually advance regional priorities that are aligned with FIP's global objectives. Presentation today will highlight the relevance of the Biopharmaceutical Classification System based Biowaiver regulatory pathway, as well as outline the regionally driven approach to implement harmonized biowaiver legislation among Latin American countries. FIP's role as a facilitator in raising awareness among relevant stakeholders can also be adapted to other priorities in different regions that are aligned with FIP's global vision of the world, where everyone benefits from access to safe, effective, and quality and affordable medicine. And so we hope that you find inspiration in opportunities for collaboration and partnerships with FIP on various topics, and in this particular session, we will address some scientific topics that are pertinent to the Board of Pharmaceutical Sciences within FIP. So with that, the various topics that are addressed here are listed and will be presented by highly qualified speakers that I will introduce in a few seconds. What we hope to achieve with regards to learning objectives are listed here. Certainly, we hope that at the end of the session, everyone will have a greater understanding of the public health implication of the BCS-based Biowaiver regulatory pathway, as well as being able to differentiate between the in vivo bioequivalence and the BCS-based Biowaiver regulatory strategy for generic drug approval. On the other hand, we hope that this webinar will give you an opportunity to recognize the value of FIP-supported but regionally driven partnership to advance universal health coverage worldwide. So the first speaker in this series is Dr. Steve Cordero from the WHO. And we're very pleased to have Steve participating in this event today because we in FIP have been a long-standing partnership with WHO on biowaivers. And Steve is currently the responsible person for leading that project. As you can see here, Steve is highly qualified with expertise in drug development, regulatory affairs, and health technology assessment. And at this point, I want to invite Steve to turn on his camera and provide his explanations about the WHO Biowaiver project. Please, Steve.
Thank you so much, Giovanni. Just before I moving on, I would like to check if you can hear me and see me, just to confirm that everything is working fine.
All good, thanks.
Thank you so much, Giovanni. So good morning, good afternoon, and good evening to all the colleagues. My name is, as already introduced by Giovanni, my name is Steve. I'm a technical officer at the WHO Norms and Standards for Pharmaceuticals team. And today I will be, I think, okay. And today I will be leading us through the Biowaiver list, which is a tool to help us reach universal health coverage. I would like to firstly before starting my presentation, to thank FIP for the invitation in the person of Giovanni, and also to thank to all the participants and extend my regards to the other panelists. It is with a great pleasure that I have this opportunity to talk about the work that we have at our team within WHO. Having said that, and now I will explain you, and this diagram explains in essence what's the work of our team in the area of bioequivalence. And it goes from the actual development of the guidelines and the regulatory standards in the context of establishing bioequivalence to the establishment of the Biowaiver project aligned with such guidelines and such requirements, so that such project develops and generates results, which ultimately will be inputs for the Biowaiver list, which is our tool to facilitate the establishment of bioequivalence of generic products. And again, my presentation today will also be structured in that way. I will start with some concepts related to the more normative work and the aspects regarding the actual establishment of bioequivalence. And then I will move on to the project and to the Biowaiver list. So with that, next slide, please, Giovanni. Thank you. So, and what is a Biowaiver in the context of the establishment of bioequivalence? A Biowaiver is an important tool or strategy used for waiving the regulatory requirements for in vivo BE studies in drug development. For instance, and here specifically, the WHO recognizes the possibility of waiving in vivo BE studies for immediate release solid oral dosage forms with APIs belonging to classes 1 and 3 according to the BCS classification system. Again, using comparative dissolution studies as surrogate proof of bioequivalence. This is exactly an extract as it is stated in our guidelines, and this is what is called the BCS-based Biowaiver approach. There are other approaches in terms of granting a biowaiver, but the one that it's more streamlined and the one that our tools are being created for, it is the BCS-based biowaiver approach. And with that, next slide, please. Again, not only the BCS-based biowaiver approach, but other biowaivers, other approaches to biowaivers, which are entailed into the respective guidelines, prove several advantages. Not only the fact of being cost-effective, again here coming to the resources into the development of studies, but also the time that one needs to use when in comparison with in vitro studies is comparatively cheaper. We also have a simplified approval process, so it reduces quite substantially the timelines to get an approval for a generic. It ultimately reduces the clinical trial. So from an ethical point of view, not only, of course, from the economical point of view, I mean, but also ethically and scientifically, there is advantages to the use of biowaivers. And ultimately, we do believe that by implementing those or by granting those within the context of a regulatory approval, we will increase or improve the access to medicines, to essential medicines, particularly in low and middle income countries, where resources are generally scarce, both from a human point of view, human resources and capacity to be able to cope with the needs, but also from a monetary or economical perspective. Next slide, please. And so to understand the BCS-based biowaiver approach, one first needs to go to the actual classification, so to the actual BCS system. And here, APIs are basically classified according to their aqueous solubility and their intestinal permeability, meaning that if an API is considered to be of high solubility and high permeability, it is classed as classification number 1, up to classification number 4, for instance, where we see that the API, when the API presents a low solubility and a low permeability. I will later on explain how do we classify APIs as being high solubility or low solubility, and the same for permeability. But this is just to put it in context. What is the actual, what do we mean as BCS? So the biopharmaceuticals or biopharmaceutical classification system. Next slide, please. And again, the BCS-based biowaiver, it's based on the BCS classification, meaning that it stems from the characteristics of character, or the characteristics at the API level, which, as already mentioned, it's related to not only solubility but also permeability. But these are not the only factors that one needs to consider when looking at the BCS-based biowaiver. We also need to look then at the actual finished pharmaceutical product level, and here there are 2 other components. Again, there are more details in the guidelines, but this is just for the purpose of this webinar to give you a high-level overview. Of course, there are other conditions and requirements, but at a high level, we would have to look at the dissolution profile and also the excipients characterization. And these 4 components ultimately, from a scientific point of view, govern the rate and extent of API absorption from, once again, just to highlight that it's for immediate release solid oral dosage forms, but also, of course, has a direct impact per se into the availability, or I mean, eligibility of a specific product to a biowaiver. Next slide, please. So how is an API classified as being of highly soluble and highly permeable? So this is, again, just a small extract from the actual guideline that the WHO has in this area. You can see here the reference, but basically, and I will now read it out for clarity. An API is considered highly soluble if the highest single therapeutic dose, so it's just to mention, it's not the highest therapeutic dose, but it's the highest single therapeutic dose, so that can be given at a given point in time, is completely soluble in 250 ml or less of aqueous media over the pH range of 1.2 to 6.8 at 37 degrees plus or -1 degree. The API, now on the permeability aspect, an API is considered highly permeable when the extent of absorption in humans is 85 or more % based on a mass balance determination or in comparison with an intravenous comparator dose. Again, this is just to put it in context, the way we classify an API in accordance to their solubility and their permeability. And this is the actual testing conditions are further described in other guidance texts. But this is just to put it in context, the way we classify an API in accordance to the BCS system, which relates to it's related to high solubility and permeability. And again, just to keep in mind that later on for the Biowaiver project, we touch more on the solubility aspect. So to keep in mind that how we actually classified as being highly soluble or or having low solubility. Next slide, please. Another important aspect is the dissolution. And again, here, as I mentioned, the dissolution profile of the finished pharmaceutical product. Just to mention that within the context of a biowaiver, it has always, so a product has always to show a very rapid dissolution characteristics. And again, not only in terms of how it is clearly defined as being very rapidly dissolving or rapidly dissolving, but also how the dissolution profile is in comparison to the comparator product, meaning the originator. So that they need to have a similar dissolution profile, and they also need to observe the definition of being very rapidly dissolving or rapidly dissolving. Here, just for clarity, one considers as being very rapidly dissolving when more than 85% is dissolved of the FPP is dissolved within 15 minutes. Again, specific, and there are specific testing conditions that need to be observed, and these are further described in the guideline. And the same for rapidly dissolving, which is instead of being 15 minutes, is 30 minutes. So again, these are components that are important to keep in mind when checking if a product is eligible for a biowaiver or not. Next slide, please. So coming back to the eligibility, and if all of you remember the definition that I presented in the beginning, which relates to an API being eligible for a biowaiver if it's under class 1 or class 3. The same now, looking from the perspective of the actual product, then we have these two classes that can direct whether or not a product is eligible for a biowaiver. Class 1, again, if the product is highly soluble and highly permeable, and if in comparison to the comparator product, the generic is either very rapidly dissolving or just rapidly dissolving. So this is again to be eligible for a biowaiver, a finished pharmaceutical product under class 1. So with APIs that are classified as class 1, need also to observe this dissolution profiling. When it comes to the excipients, which was the fourth pillar, as you might recall, the full criterion conditions are in the guideline, but it's much more lenient. So it means that there is no need of specific requirements apart from observing certain excipients which have an impact on the intestinal absorption. But again, it's much more straightforward to apply than the class 3. For instance, in the class 3, the product needs to show basically shows low solubility but high permeability, and here the product in comparison to the comparator product needs to be very rapidly dissolving. So it needs to more than 85% of it needs to be dissolved within 15 minutes. And here again, on the excipients aspect, the product formulation needs to be both quantitatively but also qualitatively the same, and in similarity to the comparator product. So this is a condition when it comes to APIs of class 3, that the FPP needs to be very rapidly dissolving, and at the excipients level needs to be quantitatively and qualitatively the same. So again, full criterion conditions are for in vitro equivalence are present in the guidelines, but this is just to put it in context that the BCS-based biowaiver approach means that if you have an API of class 1 or class 3 classified according to the BCS, it is in principle eligible for a biowaiver. Of course, observing the other aspects at the FPP level, namely the dissolution profile and the excipients composition. Next slide, please. And why do we have the biowaiver project? After presenting all these aspects more from a normative point of view, the WHO felt that both member states, regulatory agencies, and manufacturers would benefit from having a list which would present a characterization at both the solubility and the permeability level. And here, this information would ultimately direct the countries, both from an assessment point of view, but also the manufacturers from a strategic point of view, to look at those components where there could be a streamlined approval process, and therefore faster access to essential medicines in some of the countries by applying the BCS-based biowaiver approach. However, at the time, this initial list, which was basically classifying some APIs from the essential medicines list at the time, was based on literature data. So it was based on articles and other scientific data that was available publicly, which, as all of you might be aware, already poses the challenges in terms of the harmonized approach to actually testing the APIs. So of course, there was not only reason for bias, also the scientific rigorous was in question. And in some cases, as part of the assessment of these products, there was the feedback loop from the countries that the classification was actually not entirely correct, that there was occasions where a product, instead of being correctly classified, was not. Therefore, the 52nd ECSPP, and here ECSPP stands for Expert Committee on Specifications for Pharmaceutical Preparations. It's the expert committee responsible or that works within the scope of norms and standards for pharmaceuticals in the WHO, recommended or made the recommendations for us to develop a new biowaiver list, and in this case to be based on verified laboratory data. Again, the main aspect here was really to create something that could be of use for both both manufacturers and mainly for both manufacturers and regulatory agencies. Of course, there are other parties that would make use of it, as I will explain also later on, but that would be a reliable one. So that there could be not only credibility in what was being produced, but there could be that would be a factor of reliability into it. So we would have a standardized and an harmonized approach to it, which could be followed by our network of laboratories. And again, what we're basically doing is, in essence, as part of this project, is that we are assessing the APIs that are based on medicines that are listed in the essential medicines list, and we follow a protocol. So as part of the project, a protocol was created, and I will show it later on as well. Just some reflection of it. But there was a creation of a protocol that would guide the studies to be conducted under the biowaiver project, so that we could look at the classification of the APIs under the BCS, so the Biopharmaceutical Classification System. And now are we doing it? So we are doing it with the collaboration of FIP, with the collaboration of universities, quality control labs, with the collaboration of our pre-qualification team of medicines, and also with the support of pharmaceutical manufacturers. Again, here, API, especially API manufacturers, which also benefit from such list. Next slide, please. And so what is the focus of the project? Also from a logistical point of view of addressing the BCS classification, and also in accordance to the protocol that was developed. So our focus of the project is really to look at the solubility classification. So as you remember, I introduced, or I made the statement on how an API is considered to be highly soluble. So we basically, the protocol, it's around this. So it's made taking into account the regulatory standard of considering a product highly soluble, which means that as part of if a lab follows the protocol, it would be able to have as an output whether or not the product is highly soluble in 250 ml under the specific testing conditions. And here, by saying if it's highly soluble or low soluble, it means that it is either class 2 or 4 if it presents low solubility, as you might have recalled from the BCS diagram that I've shown, where class 2 and 4, the API is of low solubility, or class 1 or 3 if the API presents high solubility. And here, by having these outputs, we already know if a biowaiver is impossible in principle. And we also exclude those APIs from the ML where a biowaiver is not possible. Again, just to highlight here that there are some components that need to be observed at the FPP level to be in line with the regulatory standard, but these are just aspects that can be further consulted later on if you wish. But the focus of the project is by looking at the solubility, we can already have a division of those APIs that are either eligible for a biowaiver or not. Next slide, please. So this is the protocol. This is just a screenshot of the main page of the protocol, and the source is also in the presentation. And again, this is the scientific methodology behind the biowaiver project. So the labs observe to the protocol when they receive the samples, and therefore we ensure that there is a reliable output that can be that results, no matter if the lab is in Brazil or in the US, we make sure that the labs will operate in the same manner, and therefore the result will have observed the same protocol, which it's an important aspect to ensure that the tool, the list is reliable, or the output is reliable. Next slide, please. So this is just again another screenshot of the current our network of laboratories, so that you can all see that we have a global representation, and even here in Latin America we work with a lab from Brazil, and we do hope that we are able to also extend that collaboration in Latin America. So who knows if in the future that map could be further painted in green. But again, this is just to really show that by having an extensive network of labs, we can have not only an external control to the experiment, but also an internal control to the same. The way we, of course, operate within the Biowaiver project, where labs have a look at an API sample from the same batch, but we also have labs that look at API samples from different manufacturers, and therefore different manufacturers. So by having this control, we ensure that if there are issues with the outputs, this can be looked at on an ad hoc basis and in more detail. But again, this is more just to give you the outline of our network of labs. Next slide, please. So this is the end result. The end result with all the outputs, and I do apologize that this cannot be looked at in detail, but again, the source is also available in the bottom part of the slide. But this is the end result of all the outputs from the Biowaiver project. So through the Biowaiver project, we get the solubility outputs, which means that we are able to classify the APIs in accordance to the BCS. Therefore, if they are either class 1 or 3 and eligible for a biowaiver, or if they are class 2 and 4 and are not eligible for a biowaiver. Again, this tool that is available for a wide range of stakeholders, in particular regulatory agencies and manufacturers, allows them to very easily, within their work plan, to understand the APIs that they can, for which a biowaiver approach can be applicable. And again, from our side, as a result of the biowaiver project and this harmonization initiative to the way we look at the classification, we were also able to, for instance, as you might see, I know it's not visible, but there are some APIs where there was a change in the classification in respect to the 2006 list. Here already showing and being aligned with the feedback we received that some APIs were not properly classified. So again, this is an important result, an important outcome that only supports the Biowaiver project even further. So we are happy with that because it means that there is a reliability into the list. But also we have APIs that have been characterized for the first time. So APIs that were not in the 2006 list. Of course, we do also need to characterize APIs that have been in the 2006 list, not only to verify those, because they also need verification, but also because some of them, they have been in the market for quite some time, and they prove themselves to be, if one are to prioritize, they prove themselves to be quite essential for countries. So we cannot put them aside just because they have been around for quite some time. So they also need to be characterized. And of course, as part of the prioritization exercise that we do at the WHO, we take all of these aspects into account. So next slide, please. This is just the list of APIs that we are currently studying. So the ones that are gray shaded are Plan B, so they are not really being classified or studied this cycle. But we are having a look currently at 9 APIs, and we do hope that by October this year, we would be able to have the Biowaiver list updated with the results from these APIs. And again, this available is the source for this information is also available in our report as referenced on the bottom of the slide. Next slide, please. And how have we been collaborating with FIP? And again, I would like just to reiterate how important and how valuable a collaboration with FIP has been. And I think from the slide you can already see the wide areas of contribution that we have been receiving from the federation, namely initially from a scientific methodological point of view regarding the establishment of the Biowaiver project, the development of the protocol from the steering group, and that gives us guidance and a strategic guidance into the Biowaiver project for the next cycles. From ad hoc support, scientific support to labs. So of course, as one might imagine, lab conditions varies depending. Sometimes you have challenges depending on the API, and FIP has been crucial in providing us with support to such labs. Again, the project was established with the network of labs was also established in collaboration with the FIP. So this came into life due to our partnership with them and their contacts within this area. And ultimately, as part of the prioritization exercise, and given that the FIP has already done some work in the past in this area, we also receive crucial input from them into what APIs should be studied, namely considering those ones where there is a borderline case, whether or not they are considered highly soluble or low or of low solubility. So it's very important for us to get this input, this feedback from external partners, namely FIP, because it really guides us on making sure that whatever we produce is of use and it's relevant and answers to questions that have not been answered so far. So again, just to reiterate how grateful WHO is to the partnership we've been having with FIP under this project. Next slide, please. And in summary, and to finalize my presentation, we do believe that the Biowaiver list, so the end result of all this process, serves itself not only as a tool to facilitate the establishment of bioequivalence, but in consequence, it will result in several benefits. As a result of having this tool, this list, we will have several benefits and for different stakeholders. So from regulators, as again, some of these advantages are repeated into the applicability, are repeated from the applicability of a biowaiver, but here specific to the biowaiver list, just to highlight that this can also be used by procurement agencies. It can also be used by payers. It can also be used by manufacturers. It has an impact on the ethics by reducing the number of trials, and it also reduces the investment from a resources point of view, both from human and financial resources. Again, I will not dive into the specifics as it is presented on the slide. I'd just like to finalize by saying that with the biowaiver list, we have a tool that ultimately will enable and will improve the access to essential medicines, which is a crucial factor to reach universal health coverage. And therefore, we as WHO, we are very supportive, and we do believe that this is a great tool to be used and to be used by all of you. So please do consult it regularly, and every time we have public consultations, do let us know your feedback and which APIs would you like to have us tested. So with that, I think I've finalized my presentation, and I will hand over to Giovanni. So thank you all of you for your time, and I would be happy to answer any questions later on. Thank you.
Thanks, Steve. A great intro to the topic, and appreciate the overview on the scientific concept as well as the relevance of this biowaiver concept from a global perspective. And I'm sure there will be some more questions for you later on during the panel discussion. So please, if you have questions after the presentation, put them in the Q&A box. Now I'm going to move to the next presentation, which addresses the FIP Latin America project, which is a regionally driven project. And I'm very pleased to introduce to you Thalita Ferreira, who has been serving as the coordinator for this project for the last 2 years. And as you can see, Thalita has a wealth of experience in different areas relevant to regulatory affairs as well as health management in the Latin American region. And so without further ado, Thalita, please outline the logistics that we were using for this project.
Thank you very much for this opportunity to present again about this project that we are conducting in Latin America. And hi, everybody. Thank you very much also to the team that is participating in this project. But before to present the results that we had in these last 2 years, I'd like to present to you some challenge and difference that we have in Latin American region. We have more than 20 countries in our region with a huge population that speak 3 languages, Spanish, Portuguese, and French. Please, the next slide. So this difference that we have in our region is also in terms of political, economic, and life expectation. So we needed to handle it when we wanted to work with medicines and understand the difference that we have for each country, because each country also has a specific regulatory environment, and the population must be connected with that to introduce product with quality and safety. Next slide, please. So a study conducted in 2021 present that 100% of Latin American countries don't have a biowaiver legislation. And the countries that have already implemented a specific regulatory environment for biowaiver don't have an harmonization. So it is something that we need to think about, how we can work together in our region to harmonize the requirement that to implement biowaiver and support the different stakeholders to presented what is Steve showed to us today, that is necessary to classify the substance according what is necessary for biowaiver drugs. Next slide, please. Based on that, we connected on a collaboration between WHO and FIP to support us in the region. We understand what is necessary for implement one FIP agenda and create a community that could work together in collaboration to develop a regional roadmap. It was a challenge for us because, as I presented to you, we have a lot of difference, but also we could understand what is important for our region, which substance the government would like to work to support an introduction with drugs with low cost. It is something that all the time we need to be connected to create this community and continue sharing knowledge about biowaiver. Next slide, please. So, based on this WHO and the FIP collaboration, we created a project called FIP Labs, and that we have the participation of Giovanni that coordinates as a scientific secretary of FIP. We could connect with Chile, Cuba, and I was represented Brazil, and we could to have the different stakeholders participate in this project with three main objectives. The first one is to develop different activities, meetings, and workshops to continue sharing the knowledge. The second, to create a biowaiver regulatory pathway, or we could translate it in regulatory roadmap for everybody understand what is the difference among the different countries that we have, how we could to support the laboratories to implement the techniques that they have to prove that drugs could be biowaiver implementation. And the last but not least important, to harmonize the biowaiver regulatory approval process within the Latin American region to support the health authorities with the legislation according to the government's needs. So it is so important to work together in the region to understand how the government and the health authorities could support the pharmaceutical companies with the aspects and requirements that each drugs that would like to register, it's necessary to comply with the requirements, local requirements. Next slide, please. The meetings that we started, it was in July of 2021. We have the participation of different countries, and these meetings and the webinars that we conducted during 2021 could support Argentina to implement a biowaiver new legislation. And not less important, we also create some documents that are under revision, a white paper that describes the result and the scope of the project, and articles that also are under review to send to important journals to share the knowledge that we could acquire during the time of the project. Next slide, please. And these meetings and webinars we continue during 2022, and we have participated more than to 500 people, and not just from Brazil, El Salvador, from Latin American region, but other regions. and it was very important for us to share what we could understand about the difference that you have Biowaiver requirements. And also we presented this result in the Congress last year in Seville, in Spain, that we could connect with different other regions that are very interesting to implement the same project that we conducting in Latin America to Asia and Africa. And so with this connection and the relationship that we created during these last 2 years, we create a commitment with the participation from Chile, Brazil, Argentina, Colombia, and Mexico to continue with the experience that we acquired and to connect with the other stakeholders that can implement a new legislation for Biowaiver and understand how they can implement more projects inside their companies. Next slide, please. So as I told you, have the participation more than 500 people in the region from different stakeholders, from a pharmaceutical company, universities, and different countries. And now we would like the participation of more people to continue this project and also to implement a BCS library that it was important to classify and identify what Steve presented here, which substances has a specific solubility and permeability that could be included in each legislation that the health authority could present to their population. Next slide, please. So to conclude in this my presentation, I'd like to thank you to everybody that is participating in this community, from Alex, Andres, Eduardo, Carmen, Diego, Ervin, and Adi, but also invite everybody from pharmaceutical companies, universities, and others that are very interested in this project to continue to implement the legislation that is necessary for our region, but also continue sharing the knowledge that it is so important to include medicine with low cost, but also with safety and quality. Thank you very much.
Thanks, Thalita. Nice to have this overview and also understand a little bit more about the diversity of the different regulatory approaches that are taken within the Latin American countries. And as you explained, it's not a consistent implementation across those countries. And now in our next presentations, we're going to have the opportunity to learn a little bit more details about the Biowaiver approach that is taken in countries that have more experience and countries that just started to implement that. So with this, I'm gonna move to the next speaker. And this is Alexis Aceituno from Chile. And we're very pleased to have Alexis with us today, because as you heard from Thalita's presentation, Chile has been a country that has quite a lot of experience with Biowaiver for some time now. And Alexis has been an integral part of the steering committee and the support that we have gotten for this project. And with his experience in regulatory affairs and public health and the knowledge from Biowaiver's implementation in Chile, we're looking forward to learn more how this could help other countries. So Alexis, please.
Thank you, Giovanni. I would like to thank the FIP for the invitation to this project, as well as the audience. I'm not sure how big it is, but I'm guessing that we have so many people around, and that's very good for them to have a general picture of what we have done on biowaivers in Latin America. Well, the name of my presentation actually is BCS and dose strength-based biowaivers, because we have decided to launch this as a tool for general situation, as one of the previous speakers said. Biowaivers is not only focused on BCS classification. As a tool could be used to grant biowaivers to different other dosage forms. We actually have a lot of experience, 15 years of progress and challenge, because we started the other way around. We started with biowaivers, because at the time we started, we didn't have so many CRO centers in our country or nearby. But we do have several quality control labs. And at that time, we thought, well, why if we started changing our regulations for those facilities to be used in terms of giving us the opportunity to establish biowaivers in Chile. And saying that, the experience has been a lot of challenging first. Yeah, it's been a lot of challenge, but we have so far so many data and a lot of progress in terms of what we have learned, and we're still learning about how to apply biowaiver, how to review them, and how to get a better picture of what's going on with our formulation in our market. Next one, Ioanni. This is a little disclaimer, just saying that views expressed are mine and do not necessarily reflect official Chilean ISP policies. Next one. Yeah, and actually, as I was recently saying, our regulatory framework is very well described in our sanitary code, which is a Supreme Decree number 3. And we have several other technical guidance that describe in detail how to conduct studies to demonstrate bioequivalence. And one of them is based on BCS and those proportionality biowaivers that we already granted in Chile. Our current regulatory scope includes IR and ER solid dosage form. So what we said about biowaivers application, we said you can apply with a BCS-based biowaiver when the drug has some physical chemical properties, and the product has some specific performance. In the case on the size of the drug, we said that the drug system must be always highly soluble, and in terms of permeability, it could be highly or lowly permeable. And in terms of drug product performance, we said that the drug release rate, which is measured with a specific apparatus, must be rapidly or very rapidly dissolving. The other thing is the dosage form must be of immediate release. And still, we're trying to see if we can address a possible BCS-based biowaiver for orally disintegrated tablets or fixed dose combination waivers when one of the PA it's actually one possible candidate for BCS biowaiver. Next one. Next. As I was saying, we have guidance, specific guidance for BCS and dose strengths for proportionality biowaiver. We decided to put together this in the same guidance in order to have a biowaiver, as in general, as a tool for any kind of biowaiver in the future. And this is true because we have several editions of this guidance since 2008. As I told you, we started the other way around. We did not start it with the BE study classical that it's done in vivo with volunteers because of all the disadvantage and all the problems we have at the beginning. So we really think that having the possibility of implementing tools like BCS or in vitro approach, that was an easy way to go, an easy way to start it. And I think it being part of the project right now, that was my general recommendation for other jurisdictions that they have not advanced, because they had the same question that we have it at the beginning. We said, well, we don't have CRO, we don't have implemented, we have not implemented any clinical research guidance, etc., etc. So in that case, the possibility to start with BCS Biowaiver is a very big opportunity for just started. This guidance, for instance, describes every single detail. This is a screenshot where actually it's in Spanish, where we describe how to perform the solubility characterization, permeability, to look at the effects of the excipient, to look at the effects of some of the formulations on the performance as a measure in terms of dissolution profile comparison. And also, as I told you, this guidance, which is very popular in Chile, also is being used to grant a biowaiver based on dose proportionality. That is the case when you have at least one in vivo study, and you can compare the other strength based on the one that it performs the in vivo study. Next one. Our technical guidance to conduct BCS and dose strength biowaiver studies look at strongly and drug product formulation, because we really believe now, with all that the experience we had, that it's time to look at the excipients or compositional formula as an active ingredients in terms of biopharmaceutical properties. At the beginning, we were more on the sensibility to have a better picture of performance, maybe on the physical chemical drug characterization. But at this moment, because we have so many pathway gone, we were looking at more in detail. We're trying to dive deep in terms of what the excipient could affect the classification and could affect other properties of the drug, of the active drug, in terms of granted or not a biowaiver for this kind of formulation. So as I told you, we are right now just looking, because we have more experience and more progress on BCS application. We're having more, our focus has been more on excipient as it was in the previous years. Next one. Saying that in the guidance, for instance, is described what excipients are acceptable for class 1 drug. We usually look at the allowed usual excipients with quantity consistent with the intended functions in the formulation. For example, a lubricant, where when, for example, new excipient or unusually high quantities of common excipient for immediate release dosage form are used, for instance, surfactant, in that case, the sponsor must justify and give evidence that they do not affect bioavailability, and we ask additional data in those cases. When a sponsor uses, for instance, big amounts of polysorbate or sweetener like mannitol or sorbitol, that right now we consider that may be questionable, and we ask for more information about those changes. Those statements are for the kind of formulation where we're in the presence of formulation that have a class 1 drug. For class 3 drug, we are even more strict. They are not described completely in the guideline, and it's assumed so far that the analysis under this classification should be done case by case. But anyone will still have some hints that it's shown in the next slide, Giovanni. For instance, what are the ideas that we have in the guidance is to look at both reference and test product formula. If quality and quantity similar formulas are acceptable for reviewing, but if it's similar but quantity of excipient that are complicated for a differs, applicant should justify not impact on bioavailability, and otherwise we dismiss application. And now, right now, I would say a few months ago, we're currently moving toward an experimentally based requirement on the effects of excipient, specifically on permeability, drug permeability. In the previous period, we were looking at permeability, drug permeability, instead of being demonstrated based on literature data or in silico approach, but that information right now can be used as additional support. We really want to, the applicant should be able to demonstrate that experimentally those excipients are not anymore questionable in terms of regulation. Different approaches have been used to evaluate drug permeability, and these are described overall in the world, but in our Chilean Biowaiver guidance, we have seen light at the end of the tunnel, as just a joke, because right now we have finally implemented labs in progress and been authorized. So we have the tool. The tool is being authorizing those centers where they can, the applicant should go and can they ask for seeing if the formulation or some excipient have or not have some effect on formulation. Even more, it could be used in case of we have a classification that is ambiguous and we don't know if it's class 1 or class 2 or some other class. So we can get a better picture of real classification, as one of the exponents here said that some classification are not really clear, even with the literature that we have up to date. Next one. We also have, if we look at the guidance, we have a close look at the characteristic of dissolution release rate or dissolution performance. So we look at the composition of formulas and specifically solubility. So the GBOF, well, I'm going to say that in Spanish, GBOF02 guidance express that excipient should be critically evaluated in terms of the type and amount used. For instance, if we are talking about surfactant present in the formulation, they should be critically evaluated in terms of being permitted or not in that formula. Excipient effects on drug dissolution solubility are easier to assess through dissolution release profiling. And from a regulatory perspective, BCS drug is dissolution solubility testing is the most conservative estimate. This is the one thing that no one said every time, but actually it's really true. BCS is very conservative, gives us more information than the in vivo study, and actually technically or I would say scientifically, it's a very good tool. So it should not be understood that having a medicine authorized at BCS is going to have a less quality or maybe be doubtful. That's not true, because BCS is very conservative. So in terms of that, biodissolution solubility may differ instead of a media component and even volume, because we're always can compare if a dissolution is being occurred by actually pockets versus a bulk volume. Next one. Moving to the other tool we have described in the guidance is the test that is done when we're trying to look at the dose strength Biowaiver and dissolution testing. In vitro dissolution testing, just a reminding, plays a crucial role in demonstrated product strength bioequivalence, excuse me, for a strength not tested in bioequivalence trials. So the idea here is to use this tool as much as we could, because it can reduce it can reduce the cost, especially the cost, and the way to have the other strength being approved under the one that is being proved to be bioequivalent to the reference product. So this type of tool is very convenient, because we have it's a way that is time and cost effective alternative to bioequivalence studies. What is understood in our regulation about this type of waiver is that actually the formulation has to be the same, other than color and flavoring in agent present in our formulation. They have the same manufacturing process. The other thing is we made guess that the pharmacokinetic linearity is a model, is in the therapeutic range, or it's described in the therapeutic range, and also the in vitro solution condition that are going to be used to this kind of biowaivers. Next one. Well, this is a result, and I just want to stop on this slide because it's really nice to look at that and see that starting all the way around, it wasn't a bad idea. We have from the overall number of application, 100%, approximately 20% applications that have already been approved have been done by, as we can see here, BCS Biowaiver. 20% is a good number, it's a big number. And if we add the possibility to use the waiver tool to those strength proportionality, we can add these two together and to have near 40% of applications that have already been approved in terms of demonstrating their efficacy in our market. All these numbers we have here are real. This is not academic. It's real. This is the real world, and that's the good thing about this, because when I was in academia, we were very academia type of guy and very theoretical. But being in the real world is something completely different, and that's why I think this slide is very nice, because as you can see here from here, we have so much information that can be used if we're allowed to use or not, because this is all the information that we gather from the application that we receive at the ISP every year, every month, every day. But the most important thing is, as I told you, Talita, during the project and in all the seminar we have, is that having this tool as the way to demonstrate bioequivalence can have a big impact on the different regional market in our Latin American, and I'm going to say that way because it's our Latin America, and there's a big potential, I think, for some other countries nearby that they should start thinking in getting a better picture of what BCS can be done, and a better picture of how to implement, having our experience, how to implement this kind of tool as a way to advance in bioequivalence. Because it's better to have this than having nothing. And as Talita told, there are some countries where the advances are near zero. I mean, this is really sad to say, but some of the regulatory advances in bioequivalence in some of our countries in Latin America are very poor. I know that we have some lack of resources sometimes, but you have to be like creative, not too much, but you have to be creative instead of looking the best pathway to go through. Well, this table just represents the number of bioequivalent products we have already in the market in terms of the type of studies that we have described in this graph, in this plot. Next one. Some particular considerations for BCS Biowaiver that are granted in Chile, and this, I think this should be taken as maybe as a suggestion, knowing that we have a lot of experience on that, is that we have defined that solubility, drug solubility, should be performed on raw material. So we are asked for to perform the dissolution, the solubility determination in the drug active substance raw material that is being used for manufacturing. At the beginning, it was funny because some of the sponsors were looking at the primary or secondary standard to perform the solubility. So that was nonsense. I mean, the idea here is to prove what's going on with what you are using in your manufacturing. So we are very strict in asking for this kind of description. They have to build the pH solubility profile. If the drug, for instance, if a weak electrolyte, they can use a profiling, or they can determine if they are able to use different methods to finally get a better picture of what solubility it's gonna be after the assay. The other thing that we have done in Chile since 2008 is to evaluate the solubility in the case of proportionality. We use the authorized highest dose strength. We don't use the clinical dose. We really think we have the conviction. We are really in the position of thinking that we're looking at this dose, including the dosage unit. It's not that the one the physician is going to use, because we don't know that. Some physician may use one clinical dose, some other maybe use a different clinical dose, and that is confusing, at least for us. And at that point, we have to harmonize, but we're not really lined up with some recommendations that are coming from overseas, excuse me. The other thing that we ask in Chile is, and also it should be asked everywhere, is in terms of solubility determination, they should use a method that is validated, an analytical method that it is already validated. Instead of in terms of permeability, we asked, we look at the drug where the permeability is highly described. And in that case, if the permeability for that drug is highly described, that can be supported by literature data. When we have in presence of a drug that is it's a real class 3, or maybe we don't have a clear picture of the drug is class 1 or class 2 or some other, in that case, and also if we found significant difference in excipient in terms of type and amount regarding the reference product, in that case, we right now are in the position of as a permeability study on the face of excipient when required. This is a very good news for us because we didn't have this tool before, but this is growing every day. We are learning, the sponsor is learning, and we're also learning because this is new. And I think it's kind of new, not only to Chile, maybe to the world. The acceptable permeability assay that are described in the guidance could be cell culture, excuse me, like Caca2 cells and MDCK type of cells. And as I told you, we have 2 recently authorized research centers for primarily study, and we hope this is going to be growing fast in the coming months, hopefully. And last one, and the last one, I think this is the last one. I have some remarks here. Just telling you that a Biowaiver reduces the regulatory burden without compromising the product's quality and in vivo performance. And we have the experience to tell you that, believe us. We have a discussion about if we should use the highest strength or clinical dose to classify drug solubility. And this should deserve, as I told you, some additional discussion. I think this is not clear. We're not in the position to accept what it says in some other jurisdiction. And I think there's room to discuss that. The other thing is excipient effects. This is general for everyone, and drug stability should be looked at closely for granting a BCS-based Biowaiver. A stricter criteria should be used for compositionally proportional formula in case of class 2 and class 4. Also try to use new approach for strength Biowaiver. It seems necessary when proportionality criteria is not feasible. Sometimes it's not possible to use proportionality criteria, and in this case, probably we need some additional approaches. Well, and finally, harmonization is not to have if efforts are made on willingness and flexibility during discussion. If we're not having these 2 characteristics, it's very difficult to get some harmonization. And for me, it's really like, I would say, impressive that being a part of the world where we have the same language, we all speak the same language. This is a big advantage. We're not able sometimes to have some willingness and flexibility to advance in terms of getting better medicine, better access to medicines in our countries in Latin America. I think this is the last one, Giovanni, and I just thank everyone for your kind attention. Gracias por su atención, y espero, and I hope this slide can give you a better picture of what's going on in at least in Chile regarding the BCS Biowaiver. Thank you very much. Gracias.
Thank you, Alexis, for sharing with us the wonderful experience that has been accumulated in Biowaiver regulatory strategy in Chile. And now we're going to move on to the next and last speaker today, which is Eduardo Fernandez from the Brazilian Health Authority. And he will lay out a little bit the current path that Anvisa is taking, that is just embarking on the Biowaiver approach. So please take it away, Eduardo.
Good day, everyone. I'm Eduardo Fernandez. I work at Anvisa. Anvisa is the Brazilian regulatory agency. So we are ruling this the medicines, the registration of medicines in Brazil. And I was invited by Thalita and by FIP to share our regulatory pathway in order to implement the BCS regulation in Brazil. And I thank Thalita and the FIP for the invitation. So I would like to start saying that somehow we need to demonstrate safety and efficacy of generic medicines, and we have already the tool of bioequivalence for showing these features of the drugs, but it is also necessary to have a different approach. Basically because when we use bioequivalence tools, we use human beings for the trials. So once we can have an in vitro approach, we have some kind of ethical ethical relation, as we not no longer need the human beings to show safety and efficacy of the drugs, and we can also have less cost for products development. So to use the BCS Biowaiver approach, we will have these two advantages. And for Visa, our discussion on regarding the BCS approach started in 2009. Right? So, but actually we started a little bit later, because the first papers, the first scientific approach on this topic is from the 90s, the final of the 90s. So we started a little bit later, but then we started. And while doing the revision of the requirements for the BCS, we needed to qualify our technical staff on this subject. So we need to understand more about the permeability. We also need to understand how to perform this solubility assay. Actually, we are talking about the equilibrium solubility, which is not the same as only solubility. And we have also the tools for achieving the results. The method that we are following is named Shake Flask. And we also need to understand better the solution profile of the drugs, the reference and the test drugs, and the conditions that this solution profile should be conducted in order to give us the results we expected. Next, please. So while we were gaining some knowledge on this topic, and we end up providing some publications, some Biowaiver monographies for the FIP. Actually, they are available in the FIP website, and I have some examples for the Biowaiver monography for metronidazole or stavudine. We also have the zidovudine and levofloxacin displayed in the FIP website. Next, please. These are only some examples in some monographies that our technical staff work on this subject in the beginning of our development of the first version of the resolutions that describe the BCS approach for Visa at that time. Next, please. So after gaining some knowledge, we came up with the first version of Visa publication describing the rules for the BCS approach. This was the RDC 37 of 2011, no? Right? And in this publication, Visa described the criteria for knowing for having a high permeability drug, and also the criteria for the solubility for this solubility. Actually, at that time, for us, it was not easy to find actually was not easy to find a huge information about the permeability and solubility of the drugs. Actually, we found some information regarding the permeability, although it was not very high. I mean, the quantity was not good enough to have a big list, as I will show you. But regarding this solubility, we really faced lack of information for solubility of the drugs. As for BCS classification, it is necessary to have this solubility in different pHs. So that information was not available at that time that we create the first list of drugs that could be bioequivalent. So for building this list, we described some drugs that we were aware based on scientific literature that they had a high permeability, and we have some information, but not information good enough to conclude on this topic that these drugs had some that had high solubility. So that's the list that we had at that time with 12 drugs, and we asked the companies. We actually for Biowaiver these drugs, we allowed the companies to not provide the permeability information about the drugs, as we already had it from the literature, but we asked the companies to provide this solubility in order to confirm that the drug was a class 1 drug. Next, please. And 3 years later, we improved the list, and we had 8 additional drugs that could be bioequivalent, and the overall list had 20 drugs. Actually, we used the same criteria. We will focus at the time only on BCS class 1, and we could have some information regarding the permeability from the literature for these drugs, but we still didn't have the information on solubility. So, but again, we had some 8 additional drugs on this list. And the interesting is here that for some drugs that we add in the list, we face some problems with them. For example, we had information that capecitabine had a high permeability, and we have information that this drug could be a high solubility drug as well. So we included these drugs in the list. But as we asked the companies to provide us this solubility assay later on, we verified that although the drug has a high solubility, we verified that a lack of stability for this drug at pH 1.2. So although this drug is described in the list, we have not had any product Biowaiver which has a capecitabine in Brazil. Almost the same for temozolomide, that we could also see that this drug is not stable at pH 6.8. The drug has a lack of stability at that pH, but in this case, we came to the conclusion that the drug could be bioequivalent. Next, please. And then the final update of the list is from 2016, and we included another drug only for some some specific issues for product registration in Brazil. So we came up with a list with 21 drugs that could be biowaived, although we know that for some of them we may have problems for stability doing the test of solubility. Next, please. So, but from 2016 on, Anvisa became a member of the ICH, and the ICH was starting to discuss a big guidance with some specific rules for BCS. So at that time, we decided not to update the list anymore, and we decided to focus on the working on the ICH M9, and that became a mandatory that was published in 2019, and after that, as the ICH guideline implementation is mandatory for the members, we came up to the IDC 749 of 2022, bringing some specific requirements for the BCS biowaiver. So from now on, from the IDC 749 on, we no longer have a list of drugs that could be biowaived, but actually we are open to receive the permeability data as well as the solubility data, and we no longer follow any positive list for accepting biowaivers. So all of the drugs for IR products can be biowaived as long as it is possible to meet the criteria. And actually, we also included in the IDC the concept brought by the ICH M9 for proving the permeability, which includes this scientific literature as well as Caco2 cells experiments. For the solubility assay, the test that we have described today in the resolution is not complete, is not different for what we had in the past, as well as for the solution profile, as it is necessary to show that the solution profile at the pH 1.2, 4.5, and 6.8, and using some specific condition. So next, I think this one was the last one. So this is a very short, brief description of the regulatory pathway that Anvisa decided to have in order to proceed with the BCS implementation. This is only a summary of what we have done in the past few years. So if anyone has a question, I would be glad to answer it. Thanks a lot.
Thanks, Eduardo. And if I may ask all the speakers to turn on their camera. We have run over a little bit with time, and so we have to postpone the question answer to a different session, but I want to thank all the speakers for their contributions. I think we addressed this important topic from various ends. If I want to do a quick summary, it seems from what we heard that literature-based biowaiver approaches seem to be quite challenging, and so there is an argument for experimental assessment of some of these parameters with regards to solubility, permeability, as well as dissolution properties that need to be assessed. And you've heard different solutions that were presented by the speakers. So at this point, I want to again thank you, the speakers, for a wonderful compilation of information on biowaivers, and the participant for their patience to get through this, whether it's early in the morning or midday or late evening. And I hope you enjoyed the presentation. Please feel free, if you have additional questions, to send that to me at FIP, and we will make sure that these things will be addressed. And hopefully you'll join us in one of the future presentations. Here's the email that will be shared, and events.fip.org, where you can get some more information about future webinars. Thanks so much for your participation, and enjoy the rest of your day.
Thank you.